processes. But it's no different than digitization when you think of it. And
yeah.
Hi, I'm in California.
hello.
You wish.
I wish, yeah.
Yeah, with some sun and some beach. Yeah. Hi, Felix.
Hi, Felix.
Hi.
Yeah.
Hi, Felix.
Hi.
Yeah. So don't let us stop you because you're in a flow.
No. Right. No, I was just saying that automated systems in physiology, like sympathetic, parasympathetic, and so on and so forth, are absolutely essential to keep the brain uncluttered. But when you look at those processes in their own way, they're almost digitized. And we make a big deal of digitization, but I think physiology got there well before it. And so
yeah,
a push-pull mechanism is the same as an on-off mechanism, which is really digitization.
Yeah, if you look at well, I'm generalizing because I don't know a huge depth, but from what I've seen, looking at receptor systems, generally they come in pairs. Yeah, one antagonistic and one agonistic. Yeah.
And really, I mean, nature's all about conserving everything, so it doesn't want to repeat things if it doesn't have to. So it's a really clever way of doing it, isn't it? In other words, you take one system and then subtly change it. So the subtle change gives you a diametrically opposed process.
You
don't have to rebuild an entirely different system.
No.
And so it really is. It's quite remarkable, and it does it all the time. And well, where you do see it is, for example, one of the most profound links in biology is the link between growth and energy. And most people think, well, they just coincidentally travel together. Well, they don't. Not in biology. It's highly defined. And for example, one of the major growth factors we all have is the insulin-like growth factor.
Yes.
Fundamental one.
Yeah.
But the very fact that it's based on the insulin moiety is telling you instantly that growth and energy are now tightly coupled.
Yeah.
The other one you see is with very tiny molecules, like in our colons, we make a lot of butyrate, as animals do, which is a terrific energy source. But butyrate actually regulates the expression of DNA as well. And so you've got this very, very tiny molecule, good energy source, but by the way, it's also having a deep conversation with processes like apoptosis and cell growth and so on and so forth. And so all the way through, you see in growth and energy, there's tight linkage. And of course there has to be. I mean, you can't not do that.
Yeah.
So are we expecting more people, Gary?
I am expecting. Whether they're going to come or not is a different matter.
Right.
Yeah,
so how's the winter going?
Lovely.
Oh, horrible. -5 today.
Yeah, it's cool. It's very cool.
Cold, freezing.
Hi, Eugenie.
Hello, sorry I was late.
That's all right, don't worry.
Kirsty apologises because she's putting a big grant together and she's
okay, yeah,
talking to veterinarians and things. So
yeah, no, that's important.
Yeah.
How are you, Eugenie?
Well, I've had better weeks.
Oh dear.
Yeah, I had a cardiac ablation on Monday, which was terrific. I felt really well. And then but they wouldn't discharge me because I was anemic again, so I had yet another blood transfusion. And I rather lost the plot.
Oh dear.
I'm having these every 4 weeks and it doesn't go down well with me.
No.
Yeah.
Okay. So Kirsty won't be coming today. Have you heard from Jennifer? Will Jennifer be coming?
No, I haven't. She had an article. Did you see the article she had in the Newcastle Herald?
No.
You haven't been part of the. Richard will know about it.
I've seen it, Eugene. Yep.
Our beloved colleague Robert Clancy,
Right.
who's heavily into hydroxychloroquine
Oh, right. Okay.
and ivermectin and climate. No human-induced climate change, I might add.
Right.
I
Okay.
actually know him quite well.
Oh dear.
Ivermectin is a horse wormer, isn't it? You know, I use it to
know.
worm my horse.
I wormed my horses, and when it first came out, I thought, I'll give that to Lucy, but not to me.
God.
Yeah. Anyway, we have a politician here who's, I would say, was a pseudo Trump.
Okay.
It's the best way of describing him. And he's good at false news.
Right.
In fact, he did say the US Capitol riots were fake.
Oh God.
And so, we have one of those, not of
Right.
much importance. And in fact, I think they'll get rid of him here in New South Wales because everyone's up in arms. But they're particularly up in arms about the ivermectin hydroxychloroquine article
Right.
line. And he kept saying, Robert Clancy said this, and Robert Clancy's the expert. And Robert Clancy, unfortunately, responded in the papers saying he was an expert. And the University of Newcastle has issued a statement saying he is not a COVID expert.
Yeah. Okay. Yes.
So, yeah. So that's made for some,
Okay. Yeah.
especially as I have a friend who's a very good friend of his. And I had to tell Bill not to say a word. Yeah.
Right. So the recording's on, so that's all recorded, so that's good. It's all on record now.
Thank you very much.
But the good news is our horses are not going to get COVID.
No.
Well, I mean, I don't think I've said any. I mean, I've only quoted what's been in the papers,
Yeah. Okay.
Mm.
and I've been abused for believing in anthropomorphic climate change by this.
Oh no.
That is a true statement.
Yeah.
However, yeah.
Actually, you're quite correct around the horses. There's a rank ordering of ACE receptors and COVID that I've seen, and at the very lowest end, surprisingly, mice and rats, and at the very highest end, the humans. And horses, I recall, were about only a third the way down the track. Well, literally. And so there is so your horses will be okay regardless. So,
Yeah,
but it is an intriguing issue. The dogs, cats, companion animals, and so on and so forth. They seem pretty low on the scale as well.
mink don't.
No, minks don't. Yeah, minks are quite high. Yeah.
Okay.
So what's driving that then? What is driving the difference between a mink and a mouse?
I think it's the sequences of amino acids in the ACE2 enzyme where the SARS-CoV-2 binds to, and that would be my guess. And,
David, are you talking density of receptors or affinity for the virus?
I'll be talking about the affinity for the virus for the S sequence of the virus
Okay.
binding to the ACE2.
I mean,
yeah,
because the evolutionary advantage of ACE2 is a, you know, in different species, a different sort of
It
concept.
is. Yeah, and as far as I can see, the binding of the virus to ACE2 is not exactly where the peptidase site is. So there are different sites, if you follow me. And
Yeah.
so
Where a TMPRSS2 hits.
yeah, that's right.
Yeah.
So not, you know, humans being only one species, we're stuck with one lot of sequences, but other creatures, other mammals, where you can have multiple species, have evolved different variations. For example, the bat, there's about 1,400 different species of bat, and so therefore you've got so many different permutations in terms of in terms of those binding sequences. And likewise, you have different sequences in the dog, the cat, ferret, and so on and so forth.
But does that, when I read your thing on that, and were you saying that variants have to evolve in humans because we're the one species with the one sort of type of ACE2 receptor?
Yeah.
Because of bats, it can collect, it's got so many species, it doesn't have to mutate very often.
Absolutely, that's exactly what it is. And mammalian mutation rates are low compared to bacteria and viruses. But if you have lots and lots of different species, you achieve the same endpoint.
Yeah.
And that's why that's why we've got relatively few degrees of freedom to move around that receptor for the virus.
Can I ask Felix and Gary and Kerry, are the viruses working in your countries? Can you see an effect yet?
The vaccines.
Yeah.
I don't think we've really got enough data yet. I mean, I only see stuff in the news, yeah. So you never really know. But apparently they're saying that the R rate is reducing. But I still haven't seen anything which you know really validates the notion that the vaccine reduces transmission. Yeah. So
well, we're still in lockdown anyway, Gary, aren't we? So,
I know. So you don't know. I mean, that's the thing. There are so many different aspects to this. You don't know what if anything is actually having an effect. Because it is complex. There's multiple causes and effects going on. So you can't really nail it down to one particular factor. But we just don't know at the moment. Yeah.
Sorry, there was the the news. In the news, there was a report of Professor Segal from the Weizmann Institute, and he said that he has data which show that the vaccination in Israel has protective effects regarding a reduction of the infection rate. And I haven't read the paper, but I think it's quite a solid argument.
Yeah.
And this is, they have about 35% or more than 30% of the population already vaccinated. And they probably have the best data. And I don't know, in Germany, the situation is quite diffused because they have the South African mutant, and they have declining numbers. They have about 67 per 100,000 for 1 week. This is the incidence rate, which is the most relevant public health indicator right now, chosen from the Robert Koch Institute. And it's going down nearly every day. You can see a decline. But most consultants of the government say that there is an increase of the British and the South African and Brazilian virus, which is increasing in spite of totally reduction of the incidence rate. So they are afraid that on the 15th of May or in the middle of March, they will have a significant increase again, and therefore they decided yesterday to not to get off of the lockdown right now.
Mm.
And Austria is crazy because of there's a second, the South African virus mutant was seen in Tyrol, and there are very many people, very rich people, obviously, who came to resort places from South Africa with Learjet and so on. And they had good, good connections not to appear in the public, but then they found out that Schwarz in Tyrol, which is a second Ischgl, has a very high rate of people who are infected with the South African virus. And the Brazilian virus is not apparent right now in Austria, but now in the evening news, they said that they will shut down the border between Bavaria, which is the southern state in Germany, to Austria in order to prevent people traveling from Austria to Bavaria and carrying the South African virus.
Wow,
So this is a
interesting.
very crazy situation right now, and we have very strong snow falls and very cold weather, minus 20 degrees Celsius. In many regions in Germany, the trains are also locked down because of snow and ice. And so this is a very heavy virus probably too for local sports like those. Yeah. Did you hear me?
Yes.
It's
Did you
breaking
hear
up
me?
a bit here.
Yeah.
Got the most of the conversation though.
Yeah.
Okay.
But it's a very
Okay.
new point. The one thing the virus won't give up easily is the ACE2 target, because it's too costly. If it gives up the ACE2 target, it can't propagate. But it clearly it can obviously mutate within that target. And the reason I'm so attracted to repurposing therapeutics around ACE is you can knock out the effects of that virus on the ACE2 target by being clever around the renin-angiotensin system.
Yeah.
In other words, you
Oh,
can live.
I wish someone would believe us.
Yes.
Yeah, but you see what I'm saying, Eugenie? In other words, it will, if you go that way, you haven't got this problem of constantly chasing your mutations.
Yes, that's right.
I agree with that, but there was a paper by Louise Burrell recently that showed that ARBs and ACE inhibitors were not dangerous for treating hypertension and getting COVID. And she had some statement, and I put a thing on Twitter saying, what does this really mean for using ACE and ARBs? Because she hadn't shown a positive effect.
Yeah.
She just shown that they were safe.
Yes.
Yeah.
And I cited that paper by, I call him Marco. He's from Attequant in Austria. Is it Austria that Attequant have at home? And he'd been on a case study where they had a woman who was seriously ill, and they'd measured ACE, angiotensin 2, angiotensin 1 to 7. And they started treating her with recombinant ACE2, and she got better. And I put a comment about, well, where does this leave soluble recombinant ACE2 as a therapy? And I just got the very sarcastic comment back, well, they used antibiotics. But I mean, she's involved in a big clinical trial here of ACE inhibitors and ARBs using losartan 25 milligrams.
Yeah.
And I mean, I don't know whether Jennifer's talked to you, Richard, but I don't know why they chose 25 milligrams.
Doesn't sound a lot.
No.
And it's not tight binding, is it? If I recall, the losartan.
And
is probably the worst choice.
Yeah, I think Bevan, if he was online, would suggest other ACE inhibitors as well.
Yeah.
Well, telmisartan, I reckon, is the
Yeah,
one.
yeah.
ARB. But
Yeah.
but the thing about Marco's paper is they showed with the recombinant ACE 2 that all the profile of the angiotensin 2 to angiotensin 1 reversed,
Yeah.
and the patient got better. And I read, you know, I didn't think it was particularly antibiotic related.
Mm.
In fact, you know, she had ground glass lungs, the whole lot. So she was pretty cook.
Mm.
So yeah, while you think that the renin-angiotensin system, playing with that is a good idea, not many people do.
Yeah.
And I wonder about raising the issue of aliskiren, partly because of your cysteine protease finding, but also because it's a very powerful inhibitor of the renin-angiotensin system.
Yeah, yeah. Yeah, I think there's clearly a role for physiology here.
Talking of which, can anyone tell us where we are with writing all these articles? And because I really couldn't do anything last week.
Mm.
Have we got, I know we've got Jennifer's document, but
Yeah.
have we actually got designated tasks? Or is anybody overseeing the whole thing? Or are we just waiting for someone to do something? We're not sure who.
I think that might be it. Yeah. Yeah. So I mean, to be honest, I haven't done anything serious on this topic for the last couple of weeks because I've been focusing on system science architecture. That's been my main focus with any spare time I've had. But I know that Kerry has been doing some outreach with some people, and something has been set up there. But you're absolutely right, Eugenie. You know, if we are going to put together a series of articles in a themed journal, then we need to start getting our act together, basically. You know, to be frank.
Well, Felix and I have been exchanging information, and the more I see the exchanges, the more I see that the value of approaching this from a systems analysis, complex science way is the way to go.
Mm.
Because the literature is absolutely awash with people taking relatively thin slices of a large cake.
Yes.
Yeah, that's true.
Yeah,
Yeah.
and what we can do is say, listen, this is what the cake looks like.
Yeah. Yeah.
And I think that's really the avenue,
Yeah.
and the avenue
Yeah.
forward. In other words,
Mm.
the virus has maximized its position by destabilizing a seriously complex system in humans.
Mm.
Which really means you've got to start with the basic physiology.
You do.
Did start to do a PowerPoint, and I tried to cut bits of Gary's diagram out and use them. And that's when I lost a bit of it and couldn't get it in. I hope you fixed it up.
Yeah, it seems to be okay.
It's just a statement of one of those arrows.
Hmm.
But
Yeah. No, it's fine.
I did. Sorry, Eugenie, I did think of an image. You just prompt me to think of it before. I was trying to find something that would illustrate why this one move in a complex system is so disastrous. And it's those, if you go to a wedding and you see these champagne glasses all filled with champagne that are put together in a big pyramid, one on top of the other. And what the virus has done is taken one glass out of the bottom.
that's
And that's it. It's all over.
I mean, that'd be a brilliant thing to sort of put out as a YouTube.
Yeah, it would be.
This is what
how it
Climate
works.
has have the champagne being poured and someone put the glass out. And this is what COVID does.
It does.
As an introduction to explaining the basic
Yeah.
tenets of
That's
ACE2.
right.
I started to draw a diagram with I want to call it the American Journal of Physiology, the APG receptor and its interaction into the system. But as I say, I haven't got very far. And that chapter, as it's outlined, has got an enormous amount in it. So I might try and get some of the basics down, because that, you know, I am a basic scientist. I'm not a clinician. And and stick it to you, Richard.
No, I was just going to say that if you took the analogy further, what everyone's doing is saying, well, where precisely on the stem of the glass is the virus holding it, as opposed to saying, stand back and have a look at this mess that's been caused by taking the glass out. And that's we're in the unique position of being able to look at the entire wedding catastrophe and say this is what it looks like.
Yeah, and that might be another approach. You know, we could take all of the stuff that we've discussed so far, you know, from the transcripts. You know, I've been trying to suck it all together into categories within, you know, a coherent paper as a starting point. You know, which we would elaborate on. What we could do is instead of trying to write a paper, maybe write a coherent presentation first. You know,
that's what I was trying to do.
Pictures. Yeah, that's. You were making me think of that when you were saying it. So maybe that's what we should do. Start to put together a presentation with essentially a story, and then once we've got that story hanging together, we can then write a paper from that. If we still decide to do a paper, I mean, we could do a video instead.
That's why I was deconstructing your big picture to take elements of that from the over on the, you know how it's got research and new knowledge. I was trying to use that, and that it was deconstructing your great big picture that I actually ruined it.
You didn't ruin it, don't worry.
But it was because I wanted to get my thinking in place, because I have to build up from grassroots.
Yeah,
I'm not good at. I mean, I get there by building up, because then I get the whole picture.
Yeah.
My thinking doesn't come intuitive. Wow, that's really how it happens.
I think that's a good move. I think we write the play first, and then we can do the film later.
So. See the movie.
Okay. Right. Well, we've got half an hour left, so I think that is probably the way to go. So I mean, we need to work out the logistics of that. I mean, we can do it as I mean, I have you got stuff already, Eugenie, as a PowerPoint, as a starting point?
I've got a bit of PowerPoint. I'll pull it up and just see.
Yeah. Why don't you share it? Let me.
Well, it's not much at the moment. I've just got to pull the PowerPoint thing up.
Yeah, because as an aside, I did share your YouTube presentation with quite a few people, and that had a lot of positive, you know, feedback.
Now,
you know, with the good and the bad receptor and all of that.
Oh, the nasty but nice receptor.
That's right. Yes.
Yeah. Hang on. Share. You got it?
Yeah.
Yeah.
Yeah. Right.
Okay. This was based really on what Richard was saying. Sorry, let me go back. So that just says, you know, it's genetic information.
Yeah.
And it was for a naive presentation.
Yeah.
And then I loved this concept of 37 trillion cells. And I was trying to contrast there the simple and the complex, even in a cell.
Yeah.
And then I was trying to, I mean, this is based on your thinking, Richard. you know, the systems are organization of molecules into tissues, tissues
Yeah.
into organs, organs into integrated systems, and then finally get the fully integrated individual.
It's nice.
And then I was going to try and reverse that with ACE2, which is a bit like your champagne thing,
Mm.
but
Yeah,
your champagne thing is much more attractive.
yeah,
And then I had this, which was
Oh,
just,
yeah.
I mean, this is pulled off the web.
yeah.
This one that I've used before because it's revolting. And this is where I started to, this is what I was doing
Oh, nice.
from your stuff.
Mm.
And then, sorry, I'll go down. And finally, that's from one of my papers.
Yeah.
That's from one of my papers. That's from that talk.
Yeah, naughty and nice.
Yeah, and that's
So Jenny, could you just step back one to the pathways, the angiotensin pathways you've got there?
yeah, that's
Yeah, that
Yeah.
one there.
yeah,
I mean, if you're a design person, you looked at that and you could say, what could possibly go wrong?
yeah. Well,
I
what's
mean,
it? What's
That's what the virus has done. It said, this
Wow.
is exactly what I need.
What's interesting, Richard, is ACE2 is the target for SARS and coronavirus,
Mm.
and APN, which is DPP4, is the target for MERS.
Ah, wow, I never knew that.
So basically, I think it's APN. So basically, all of them target the renin-angiotensin system.
Yeah.
Yeah.
And not surprising when you understand what the renin-angiotensin system actually does.
Yeah.
Yeah.
So, but I don't know. This is complex because, you know, you've got all these other angiotensins in here.
Yeah.
Yeah, I think
It's not that
Right.
complex, really.
yeah, I guess
Yeah.
the systems
I think we have a fundamental problem that people constrain, if they even know about the renin-angiotensin system at all, they constrain it to cardiovascular disease and blood pressure. And
exactly.
there's a hell of a lot more going on than that. This is
Yeah.
a fundamental system for managing our body's integrity. Yeah.
Yeah.
And the viruses mess with it because
Mm.
you know you can what they do is they target things that have an association with growth, so that promotes replication. But also what they're doing is they're hindering our body's response to healing and wounding, and as a consequence we get chronic inflammation, which suppresses the immune system. So it's a win-win situation for the virus. Promotes
Yeah.
replication and suppresses the immune system. I mean, it's a perfect target. It's a real, you know, it's a weak link in our armament.
Well, it comes back to our vulnerability being our complexity.
Mm.
And as I said before, the lethality of a virus is the simplicity. But if you look at the cardiovascular area, in some ways almost the marketing of cardiovascular disease is lagging behind where the science is.
Mm.
And cardiovascular disease quite appropriately started off in a mechanistic way of understanding fluid flow and particularly around blood pressure studies and so on and so forth. But the reality is now people are understanding very clearly that it's intimately linked with inflammation.
Mm.
Yeah.
And the inflammation has been driven by angiotensin.
You've got to have the biophysics, though, Richard.
Yeah, you do.
Yeah.
But in terms of going to Gary's point of the master switch in the whole game, the angiotensin and the inflammation in the cardiovascular setting is just so critical.
Yeah.
But when you get to inflammation, you have another process that follows, and that's the process of remodeling.
Yes.
And again, angiotensin pops up as being at the master switch in remodeling. In other words,
It does. Yeah.
once you've done the damage with inflammation, and you hope that the damage to the invader is less than the damage to yourself,
Yeah.
you've got to repair the mess.
Yeah.
And so,
Mm.
and that's where you get the fibrosis in tissues,
Yes.
and
Yeah.
particularly, so on and so
Yeah.
forth, driven by angiotensin too. So you're absolutely correct in saying that the angiotensin is such a master controller of these processes, and therefore vulnerable.
Yeah, and I haven't read the literature over recently over the last sort of 10 years or so, but when I was writing my last paper on this topic, I found some references that explained that the upregulation of the AT2 receptor and the MAS receptor was dependent on local release of insulin-like growth factor by fibroblasts.
Yeah.
Yeah, so the healing and that tissue regeneration is somehow connected to the growth responses that are associated with renewal.
Yeah.
Yeah.
Actually, if I could tell you something interesting, I've spent many years studying the renin-angiotensin system in the fetus,
Mm.
and the vascular receptor in fetal blood vessels, except for the umbilical artery, is predominantly 82s until late gestation, and it starts to, the 82s go down and the 81s come up.
Mm.
Mm.
And that, of
Mm.
course, fits with growth promotion,
Mm. Yeah.
and really not consolidation.
Mm.
And we mustn't forget, of course, that their use in diabetes is well to treat diabetic nephropathy is pretty well established. And I think they're even of use in diabetic retinopathy.
Yeah. Yeah.
Yeah.
So the reason why I mentioned this mindset issue is because we really are dealing with a social issue here. You know, it is essentially a culture change. Yeah, a recognition that actually this system is of vital importance to our overall health, far beyond its existing recognition. And from understanding that leads to a whole host of new possibilities in terms of how we could deal with complex diseases.
Yeah.
And this has been neglected for, the last, 15, 20 years, to be honest.
Mm.
Or longer than that, because
Okay.
I was in a conference in New York in 1980, where Captopril was. Yeah, thank you. I've been around for a long time. When Captopril was first sort of, look what we've got. We've got this brilliant antihypertensive.
Yeah.
They were giving so much buckets of it and getting agranulocytosis because they were so enthusiastic. And somebody said, well, we don't need to learn any more about the renin-angiotensin system, do we?
Mm.
We know it all. We know all that we need to know. And that's been a prevailing opinion,
Yeah.
really, since that time.
Yeah.
And getting, when I went back into research in 2003, it was to look at the properties within organs and tissues of the renin-angiotensin system, in particular the reproductive tract, because human amniotic fluid is just loaded with renin.
Yeah. Yeah, it's amazing, isn't it? Mm.
May I ask some basic questions?
Mm.
One question is the angiotensin system is not the only one which is responsible to fibrosis.
Mm.
Is
I
that
agree. I agree.
Mm.
okay? I know from a clinical point of view. I know the liver cirrhosis in alcoholic patients.
Yeah.
And this is a complex thing where you also can say, okay, there's a hypertension component and this and that. So this is the problem on analysis of complex molecular networks in the body, where you always find correlates and you don't know who is the major player.
Yes. Yeah.
So I think talking about fibrosis should be really focused for a while on fibrosis and looking what kind of components like this AT1 receptor or other let's say, pathways are generating the fibroblasts or activating the fibroblasts. The second question is going to the RAS system, renin-angiotensin system. Sorry for my bad English. What is the basic opponent who is, reducing blood pressure, so to say? So on the endocrine level, and for instance, on the nervous system level, it's just the parasympathicus, which is just reducing the blood pressure by reduction of the vasoconstriction. So also the different targets or effects of the angiotensin system with via the AT1 receptor has some opponents in other subsystems of the organism, and this is what I wanted to highlight with this kind of mobile, which Richard knows, and I'm a little bit too curious to maybe the next time I could present it. What I'm thinking from the whole organism.
Mm.
So and there was Alion Pollock, and she wrote, thank you, Carrie, I think you moderated this connection, and she wrote an article in a British Medical Journal, a brief letter to the editors. I had no time to read it exactly, and she was saying we are, in essence, she was saying we are not caring enough about the asymptomatic persons. And so the clinician, I mean, some people had COVID and didn't know it. And some people, young people, had some, very severe diseases and died. So the basic question, which is, I think, always interesting for the whole public, even if especially if the mutants are coming up, the question is, what is the protective mechanism in the organism of the asymptomatic persons? And I think, for instance, if a patient now has, for some days, he has COVID and is at home, and for some days he's short of breath, he is anxious, so he has a high sympathetic level, and noradrenergic hyperactivation, and also if it lasts for several days, and nobody wants to go to the hospital knowing that he's probably devoted to be dead. He also has a high cortisol level, probably. So, but the clinicians do not test these parameters because they have no hypothesis. They just test the usual parameters like electrolytes and this and that. So the routine parameters, and there are some guidelines for what to measure if some at the admission of COVID patients in the clinic. But they have no theoretical framework, at least a hypothetical one, which is saying this is the essential mechanism of the pathophysiology, where the RAS, I don't know, the RAS system, has the guiding role, of course, as Richard was saying with the glasses, but there are other glasses too on the same basic level. And my interest is what is the role of the nervous system, especially the autonomous nervous system, and it's starting with sneezing and the running nose and all these things. So the nervus vagus, the parasympathetic nervous system, is hyperactive in the stage of infection on the very, very first compartment. And then the question is, what is the endocrine system doing and the immune system doing? And and what is the role of the RAS system in all these three other players? Because it's part of this and that other system, of course.
Mm.
So there must be some. At least I'm interested in the holistic position to say, sick is only the host,
Yeah.
not the virus, and the host is an organism consisting of several interdependent subsystems with certain functions which we cannot define precisely. Yeah,
Yeah.
and this is what maybe you have some comments on it.
Mm.
What
Would
is the opponent of the RAS, so
Yeah.
to say?
Would you mind if I shared a slide?
I'll switch off stop share.
So this is can you see it? Yeah.
Yeah.
Yeah, so this is from the sepsis talk that Bridget and I did back in 2015, and what we did is we produced a model again in Insight Maker, Felix, and what we showed the progression in the states of health through inflammation, through systemic inflammatory response, sepsis, severe sepsis, septic shock, multiple organ dysfunction. And the pathway of COVID is similar in this regard to its severe state. Now, I'll switch on. I mean, what we were really trying to get over here was that health has a huge dependency on what you eat and also the microbes that live within you. Yeah, in terms of maintaining your immune system and your immune responses. You know, your biosystem integrity, if you like. Now, coming back to your question, Felix, in terms of how our body responds to stress, it's at several different levels. Yeah, it's at a cellular level, and how cells respond to stress in terms of oxidative, hypoxic, glycemic, and physical stresses, and that results in an inflammatory response from an individual cell. What you also have is you have this renin-angiotensin system at the local level and also at the systemic level as well. So you've got the AT1 receptor promoting a stress response, and then you've also got the MAS receptor and the AT1 receptor promoting tissue regeneration. So there's this balance going on at a tissue level. And then when you scale that up to an organism level, you've got the efferent vagus nerve and the afferent vagus nerve, again having a role to play in terms of the release of cytokines and what's the word? Yeah, hormones, or the endocrine the endocrine system. Yeah. So you've got all of this stuff going on at a cellular, a tissue level, and at an organism level, all trying to create some sort of balance, but it gets out of balance because the virus is basically pulling out that champagne glass at the bottom.
I would put in that diagram, I wouldn't call it the efferent vagus nerve is inhibitory on the heart. Okay. I would put, you've got to have sympathetic nervous system going down to celiac ganglion.
Okay. Yeah. I mean, this was my layperson's attempt. Yeah.
The point being that there's a yin and yang between the vagus and the sympathetic nervous system. And very importantly, angiotensin in the brain uses the sympathetic nervous system as a conduit to changing blood pressure. It acts in the hypothalamus. It acts systemic angiotensin. That's that angiotensin produced in the body gets into the brain via the area postrema.
Wow.
And affects the sympathetic nervous system, the dorsal nucleus of the vagus.
Right.
Is also an intra-brain renin-angiotensin system, which people like Roger Dampney have done a lot of work on.
Wow. Okay.
So basically, taking what Felix said, and his interest in the interaction with up here.
Yeah.
I agree with you. It's amazing to me that the first symptom of COVID is often loss of smell,
Mm.
Yes.
which says to me the virus is getting through the cribriform plate into the olfactory nerve.
Wow.
And it ain't so far up from there up to the hypothalamus,
Yeah.
which is a big fat endocrine control system
Yeah.
with the pituitary hanging off it,
Yeah.
and then has pathways back through down to into the brainstem and out. So I think Felix has pointed out to one system. I've never seen anyone talk about the virus getting into the brain and affecting the brain renin-angiotensin system or affecting sympathetic pathways.
Yes.
Mm.
Yes.
Well, that would be
Exactly.
great. You know, if we could tell that story, I mean, that corresponds with, you know, many of the statements that Richard's been making of, you know, taking the perspective of the virus and charting its course in terms of its interactions with, you know, our various systems. If we could make that, and you've made a start, Eugenie, with that diagram showing the pyramid. Yeah. Could we show how the virus is not just affecting the lowest level, it's affecting every single one, yeah, through its course. That would be a great story to tell.
So, Gary, I wonder if we could go back to a really critical point that Felix raised, and that was the interplay between, for example, angiotensin and the nervous system. It's a great model because there are some fundamental things that RAS, the renin-angiotensin system, looks after. One is response to inflammation. The other is maintaining blood pressure. In other words, these are vital things in the body that you can't risk not having looked after, and that's delegated to RAS. Now, so if there is a hemorrhage, for example, in a human, RAS will kick in, and it will kick in to ensure that blood pressure is elevated. But if you now start to look at cardiovascular disease, you see the following, and it's exactly on your slide. Cardiovascular disease is a disease of inflammation, a very slow form of sepsis, if you like. The characteristic of sepsis is a dropping in blood pressure, and it comes through nitric oxide production. And so a response to that, in my mind, would be a renin-angiotensin system response. We cannot afford to have blood pressure dropping. We'll be unconscious, and that's all over Red Rover. So what we do is we now turn on the RAS, renin-angiotensin system. It can work acutely by promoting constriction of blood vessels. But what it does in the long term, in the chronic sense, is it remodels the entire vasculature, including the heart. And it remodels it in a way where the lumen of the blood vessels is now smaller than it was before it was remodeled. It's not increasing a number of smooth muscle cells at all. It's just orientating their position in space. And that's driven by RAS. And when someone takes an antihypertensive, what they're doing is slowing down that remodeling process. Now, if you then come to the sympathetics and say, well, where are they? Well, they're all localized on the outside of the blood vessel. That's how they get their amplification. That's how they work. And so if you've now remodeled the vessel with a smaller lumen, the same sympathetic nervous system will now have a much more dramatic effect on blood pressure. And so you can see this interplay now going on between inflammation, RAS, structure, and nervous system. And Felix, you were quite right in raising the cardiovascular system as being an example of where all these multiple things can come together. And so if you use that same model into the lung and inflammation, you can follow exactly the same pathway. In other words, the first thing is this explosive response of the innate nervous system to the virus. And then the consequences of not having RAS acting appropriately then takes you down this other other devastating pathway with some individuals.
Mm.
The other thing I'd like to add is the following, and that is, if you go to a high level and say, what are the key transformation molecules in a human? Well, there are three: peptidases, kinases, and lipases. And RAS would be one of the kings of the peptidases. There's no question of that in my mind. But the same story applies to the others. In other words, there's a hierarchy there. There's something like 600 kinases regulate your replication of cells in the body. I'm guessing it's probably about 200 of them have the potential to alter the replication of a single cell. Lipases will determine what's on your membranes in terms of receptors and everything else. So that really what, part of the champagne glass, and go back to Felix's point where all these things are interconnected, you could start from lipase, peptase, kinases. These are the elements of what being a human's about. And then you break down to the next level in terms of the interconnection between these things. And as soon as you get to organs, when you do that interconnection, you're now starting to look at nervous system versus remodeling and structures and so on and so forth. Do you see where I'm coming from? Yep.
We had the interesting one of our research topics was comorbidity between depression and diabetes. And so we know what is the neurobiological or neurochemical constellation in case of depression. That means a low level of noradrenaline or norepinephrine, low level of serotonin, low level of dopamine. It cannot be measured directly, but we have several pathways of evidence to have this hypothesis, and medications work. And then the question was how to bridge the problem of diabetes, like insulin resistance and all these cues of internal medicine. And then we unfortunately COVID came and was destroying our group or distracting everybody in his job. And we came to the view that the pancreas is not only producing insulin and glucagon, which are opponents to each other, with I would always say asymmetric relationship. They are not symmetric antagonists, because otherwise they would be zero. But have some different kinetics and those response relations and so on. But they also produce GABA, and they respond strongly to acetylcholine and to noradrenaline and so on. So they have they even produce locally, the alpha cells, the beta cells produce neurotransmitters. And I was trying to collect data, literature, experimental animal data, who show what is going on in the pancreas on the between the alpha cells and the beta cells regarding the reciprocal control on the very local level of the pancreas between glucagon and insulin, and with the target glucose level in the plasma. So this was the fascinating thing. And then we also looked at the role of the immune system, the interleukins, who also interact in the process of developing diabetes and going down, as Gary said, to the microbiome in the gut and so on, and trying to identify the pathways. And the important point is not to go to this big data view where we say, okay, we just take all data, we generate some equations or something like that, or make some correlations and let the machines run for 1 week, and then we look what's the result. No, we try to develop a mechanistic model.
So Felix, the example you gave is a brilliant example of repurposing. If you go back and say, where did antidepressants come from? Where were their origins? Their origins was in tuberculosis. And there were drugs called the phenothiazines, like chlorpromazine and the rest, that were used to treat tuberculosis. There was a clinical observation made that patients who were treated with the phenothiazines had elevated mood. They were quite happy. And from that observation came the understanding that phenothiazines were monoamine oxidase inhibitors. And the monoamine oxidase inhibitors lifted up the noradrenaline levels in the brain and so on and so forth. So depression has a beautiful link to infection in terms of drug development and going almost in the opposite direction to what we're saying now with RAS and antagonists. But it just illustrates that the history of medicine is just full of these transitions from one area to another.
Yeah. Okay, so we've got some great conversation there again, some really detailed conversation. I've been recording it again, so I will get a transcript. I'm aware that Kerry hasn't had a chance to say very much about what's been going on with systems dynamics and her contacts. So I think we've established an important potential relationship collaboration, at least a starting point. So maybe Kerry, you could say a little bit more about what you've been doing.
Yeah, I've shared a page actually in the chat where I've just been accumulating links in terms of what's going on out there in the world using system dynamics and agent-based modeling to understand COVID. And there are some common themes really. I mean, a lot of the models don't really have the feedback that you'd expect to see. They tend to be quite linear still, which was a bit of a shock to me. And also it's only some of the very recent models that are including vaccinations and looking at the impact of that. But you'll see there's a whole range of links. And I mean, yesterday I sat in on a webinar where there's been a System Dynamics Society initiative to get children all over the world modeling COVID locally in an app called Silico. And that's very interesting. And some of the findings coming out of that were really interesting, of it being entirely a local thing, whereas some of the other models I've seen have been, on a national scale, and they were saying this is completely invalid. And we'll see what happens, because I've scheduled a meeting with some of these people. I've reached out to quite a lot of them to say, can we talk about your models? Because I don't want to start building a model if there's something out there that we can just use or add to, or if the papers are not up to date and there's something that's going on that I'm not aware of. But I put everything in that page, so if you want to take a look at any of those, you can.
Right. So that's on the Gary Big Picture page site, is it?
No, I just put it in the chat.
Yeah, we need to find a way of putting that on,
right?
So that people can get to it. But yeah, you can get through it to the chat. I mean, what you could do, we could resort back to email as well. You could send it via email as well, Kerry. For now.
All right. Okay. Yeah, I can do that. Yeah.
Oh, I got it. Yeah. All right.
Tell us a little bit more about Alison that you've been in contact with, Kerry, and the meeting you set up with Felix.
Yeah, well, that came through other contacts in my network who said that Alison Pollock is doing a lot of work in this area and has a lot of media attention, which was why I reached out to her. So hopefully myself, Felix, and her will have a conversation next week, and we'll be able to explore how we can better, work together. Okay. But she's interested in, the bigger public health picture as well, rather than treatments and vaccinations.
Okay. Have you heard of her before, Richard Eugenie, Felix?
I haven't.
Tell us a little bit more about her, Kerry, if you can.
Well, I don't know any more really than you. I'd have to go to her site and tell you more. Okay.
Okay.
It was just a recommendation. So
Right. But she seems to be quite well connected.
yeah,
Yeah,
that,
she criticizes the UK health system. She's also very familiar with the German, at least with the Robert Koch Institute. She follows up their publications,
Mm.
and she's I think she has written with some other guys an article which is very similar to an article which I have on my homepage
Yeah.
on the language, the known unknown.
Yeah.
And that's really a very epistemically critical position.
Mm.
And I like it very much, very precise thinking. And as Carrie was saying, she's mainly a public health expert. I mean, so
Mm.
she's not interested in the physiology, but in the logics. How can we explain our epidemiological problem models adequate or not? And this is my criticism also a little bit to the MIT group, because they just yeah, they have a good background, and they look for data, and they have the SIR model, and there are so many models derived from this SIR model, which they use in Germany, for instance, for the Robert Koch Institute, and for the consultants. They are used by the consultants for Merkel, for the Chancellor Merkel. And there's, I mean, they are not sociologically and psychologically based. They're just data based, more or less, and only data based. And I think they should be also based on qualitative observations. What are doing people in the public, and what is the life world of people? This is the sociological theoretical question. If you make an analysis of collectives, of populations,
Mm. Mm.
not we make models with the data which we can obtain from the people, and not are these data good indicators for the state and the processes of the population. So this is the significant difference. There's no, there are only physicians and mathematicians who are leading and very good in making this data analysis and making models, but they are not so well trained in qualitative empirical social research, and they don't know the difference between stratification and milieu, for instance, which is very important in sociology, and subjective, objective. And so there's a huge discussion in theoretical sociology how to understand collective behavior, or the relation of individual and society. What is society? How you define society? What is social? How you define it? What there's no spatial geography and sociology. There is no link. I mean, very weak links, you know, between the and so on and so forth. So these are the issues which I would raise as a sociologist, which I've studied and made a doctorate in sociology, and therefore, yeah. I have seen so many models on the epidemiological level, and they just say if the data are not valid, I mean not reliable, if you cannot repeat, and if they have this delay delay, you don't know 5 days or 7 days what is the reality behind this data, you know. You see the data on Wednesday, and you know probably what was happening on last Saturday. So then you make models with a high variation of the data already on this level, and then you make these predictions or these forecasts, and you know the weather forecasts with all these many data, and you have theoretical background with thermodynamic laws. And third, you have the supercomputers, but in epidemiology, you don't have really laws in social, like social science laws, how to explain all these things and so on and so forth. So sorry that but I think it's very helpful, because this, what I say, is something which can happen in the future, where you can bring together mathematicians, physicists, and sociologists and psychologists, and say, let's make together a transdisciplinary based
Yeah.
model of the
Yeah.
epidemic. Yeah.
Yeah.
This is not possible because of the institutional blindness and the there's no connection, no institutes for interdisciplinarity in that respect. But so we are happy, and I'm very grateful, and I will follow this. Thank you, Carrie, for this information, and I will just check it again. And they're good guys. I mean, I like them. There's Jack Homer and others, and
But and the other thing that is quite clearly missing from all of them is they put really tight boundaries around the system, and they don't consider broader impacts on the economy, on other types of health, and there's nothing about any of that, and that is a massive gap.
So we need to draw this to a close, but Eugenie, could you attempt to upload your presentation as Google Slides in the shared drive? If you don't manage to do that, could you send it to me by email, and I'll put it on for you. You're on mute, Eugenie. Would you be okay to do that? Make an attempt at uploading it.
Sorry, I just wanted to fiddle with it and make it
Sure.
smarter, but I can put it up if provided everyone sees it as crude, rude, and
No,
unfinished.
we're applying an agile technique, so don't worry. No, we're all working as a team with crude and rude stuff.
Yeah. Okay. Well, I will. I'll do that.
All right. Well, thanks a lot. That was a great conversation. I didn't get the last one transcribed. I will do that. I've got a friend who does it for me, and I didn't really want to bug him, but I will do it now. Thanks a lot, everybody. And
Can I just say?
Last thoughts.
If I put that up, can people modify it?
Yes.
And add to it,
Yes.
so that it starts as a stem.
Yes. That was the
and
idea.
get a little bit more sophisticated.
Yeah. So we can all do some work on it once we know it's there, and we can.
Okay.
You can advertise it by email that it's there. You can send a link to where it is, or I can do that if that's not possible, immediately obvious. Right. Any other last thoughts?
No, just stay safe, everybody.
Yeah.
Okay.
One question: do we have an agenda for the next meeting? Because I liked very much the presentation of Eugene. Though there was an idea just to give a brief impulse input for 5 or 10 minutes, and
Okay.
so we could discuss.
Sounds like you're volunteering to put together an agenda, Felix. Well done. Okay, and if people have thoughts about what they would like to do next time, then send a note to Felix. Thanks, Felix.
This was Jennifer's idea.
Okay. Too late to blame her. You were here. All right. Take care, everybody. Great talking to you.
All the best,
Bye.
Sarah. Bye-bye.
All the best. Bye-bye.
Okay.
Bye bye.